That will help you. See, this I have told you, syllabus pack is your reference. Since you have not made that very well or you have done it, okay, it's okay. I'll give you those 218 topics. Look at the topic. Just I'll give you an example. We'll spend 5 minutes on this exercise, okay? Let's say cell structure and functions. Cell theory and cell as the basic unit of life. Who gave cell theory? Cell theory was given by Leader and Schwann. Cell as the basic unit of life. That's all. Cell is the building block of life. Present in all organisms except viruses. Viruses are not considered living organisms. That's all should come to your mind. Structure of prokaryotic and eukaryotic cell is the next concept. Quickly you should recall what are the differences. Prokaryotic cells, no organelles. Eukaryotic cells have organelles. Prokaryotic cells do not have or the difference. Tell me the differences. Is there any difference in the cell membrane of the prokaryotic and eukaryotic cell? Prokaryotes, bacteria, cell membrane is made up of same phospholipid. No phospholipid, same in eukaryotes also. No organelles, organelles are present. Okay. Reproduction. No true sexual reproduction, though there is sexual reproduction, kind of reproduction. That's all. So the differences should come to your mind. Plant cell and animal cell, visualize. What are the major differences between plant cell and animal cell? Cell wall, cellulosic cell wall, vacuole, chloroplastids. Cell wall, vacuole, plastids and centriole difference. Okay. In animal cell, these are absent. Cell wall is absent, vacuole is absent. Chloroplastids are absent. That's all. Cell envelope, cell membrane, cell wall is the next concept. Cell envelope is anything that surrounds the cell. Cell membrane, common for all organisms, phospholipid. The nature of proteins might differ. Cell wall, present in some organisms. You should quickly recall what is the cell wall made up of in different organisms. Bacteria, starting from the lower ones to the higher ones. Bacteria cell wall, peptidoglycan, N-acetyl glutamic acid, N-acetyl uramic acid, N-acetyl glucosamine. Then fungi, chitin. Then you have some special cell walls, diatom. Diatoms have some kind of special cell wall in terms of structure, overlapping cell walls. Silica infused cell walls. Plants have cellulosic cell walls. That's all. When you look at that, these things should come into your mind. If something is getting stuck, look at only that concept, come back. So for revision, if you have to learn everything, if I have to discuss all of these once, it might take about 10 to 15 hours. 10 to 15 hours. While you are revising, you look at it, whatever is not getting recollected, go back, learn, come back. You might not need even 40-50 hours for revision. About 20 to 25 hours for you to strengthen on your weak spots. Okay? I can get this prepared for chemistry and biology also, or chemistry and physics also, for quick revision. This is the quick revision manual. So how many pages? 8 pages, 9 pages, that's all. 9 pages for you to tick the boxes. Then it is cell organelles, structure and functions. All organelles are listed. Endomembrane system. Endomembrane system includes what? Endoplasmic reticulum, Golgi bodies, lysosomes, vacuoles. That's all. Then each organelle goes on like that. So I'll do a little bit more tweaking of this and share it. Physics and chemistry also I'll get it prepared. I'll tell my staff to prepare it. Maybe by Monday or Tuesday I'll get the physics and chemistry outline for revision. Employ this for entire revision. Alright, 52, paper 52 will do. So your linear notes revision, quick recall based revision. Linear notes is you are going through everything that you have prepared. Recollection based on the framework. Paper based mastery based on papers. So you can split your time. About 2 hours for your perspective notes revision about 2 hours for running through the outline and trying to strengthen the weak areas. Rest of the time for paper-based strengthening. Quickly going through all the papers and strengthening the concepts where you are finding weakness. Question 91, exclude. Diffusion pressure. What happens when What happens when a formalin preserved filament of spirovera is placed in a hypertonic sugar solution? Now placing something in a hypertonic solution will by default lead to loss of water from the cell. The cell will lose water. But in a hypertonic solution, the cell will continue to lose water until all of its cytoplasm is lost. So what do you call that? What do you call the complete loss of cytoplasm from the cell because of the cell being placed in a hypertonic solution? Plasmolysis. Correct? Cell loses all water due to being placed in a hypertonic solution. Plasmolysis. So it is plasmolysis. It loses turgidity, it gains turgidity. Loses turgidity is also correct, but it is partially correct because the hypertonic solution will continue to pull water away from out of the cell and the cell collapses. Will the cell collapse in this case? Will the entire cell collapse in this case? Why not? If we place an animal cell, if we place a small animal tissue of equivalent size like spirovera in the same solution, the cell collapses, but spirovera doesn't collapse. Both of them are plasmolysed, but spirovera doesn't collapse. Why? Come on, common sense. I've given the clue also. Same hypertonic solution, the osmolarity of both the solutions is same, hypertonic. In one you have put an animal tissue, another you have put spirovera. The cells of the animal tissue both of them gets plasmolysed, but this animal tissue collapses, spirovera doesn't. Why? Cell wall doesn't allow the cell to collapse, but plasmolysis happens. The entire crumbling of the tissue happens. NEET genes occur in Rhizobium, Aspergillus, Penicillium, Streptococcus. What are NEET genes? What are NEET genes? NIF. Nitrogen fixation genes. NEET genes are nitrogen fixing genes. See, I have told you, in bacteria, there are 3 unique genes that will be present on plasmids. If the genes are present, they will be present on plasmids. What are the 3 genes that are present on plasmids which will give advantage to the bacteria? One is NIF gene whenever it is present. The other one is the gene which will give the ability to fertilin, F gene or fertility gene. Conjugation is made possible by the presence of F gene, and the third gene is antibiotic resistance gene. 3 genes which give distinct advantages to the bacterium whenever they are present on the plasmid, and these 3 genes are antibiotic resistance gene, which is the R gene, F gene or the fertility gene, which provides ability to conjugate. Conjugation ability is provided by F gene. NIF gene or nitrogen fixation gene provides the ability to fix atmospheric nitrogen. Presence of NIF gene makes it possible for the bacteria to get into a symbiotic relationship with something else. Symbiotic relationship is possible, increasing its survivability. Protection. If the bacteria is given shelter inside a bigger organism, the bacteria can survive better than surviving in the external surroundings. So NIF gene occurs in which of these 4? Rhizobium, Aspergillus, Penicillium, Streptococcus. It is in Rhizobium, because rhizobium is a symbiotic nitrogen fixer. Symbiosis of rhizobium is with leguminous plants, root nodules of the leguminous plants. Streptococcus also is a bacterium, but it is not involved in nitrogen fixation. Aspergillus and penicillium are fungi. Which of the following statements about the absorption spectrum is correct? If you look at the options, the regions are mentioned. Blue region of the spectrum, red region of the spectrum. So that is the spectrum of visible light. And chlorophyll A and chlorophyll B is provided, given. And peak is mentioned. Peak is the absorption peak. That is the particular pigment absorbing light at what wavelength. So wherever the particular pigment is attaining a peak, it means that in that particular wavelength it has maximum absorption. So we have chlorophyll A and chlorophyll B. Chlorophyll A is the primary pigment. It is the main pigment for photosynthesis, for light absorption. Which end of the spectrum is chlorophyll A more efficient? Chlorophyll A is a part of both the photosystems, but chlorophyll A is the chlorophyll A. What wavelengths does it attain its peak in? 680 in photosystem 2, 700 in photosystem 1. Alright. Chlorophyll A attains its maximum absorption in the red end of the spectrum. Towards the blue end of the spectrum, chlorophyll B has a better absorption than chlorophyll A. Chlorophyll B does not absorb drastically higher than chlorophyll A, but chlorophyll B's absorption capacity is slightly higher than chlorophyll A towards the B end of the spectrum. Towards the blue end of the spectrum. Now, blue end and red end, which one is of a higher wavelength? The blue end or the red end is of higher wavelength. Red end of the spectrum is of higher wavelength. Okay? So now look at the options. In the blue region, the peak of chlorophyll B forms at a lower wavelength than the peak of chlorophyll A. Is it right? Wrong. Peak of chlorophyll B in the blue region is higher than the peak of chlorophyll A, because I told you chlorophyll B has better absorption towards the blue end than chlorophyll A. So option A is ruled out. In the red region, the height of the peak of chlorophyll A is more than that of chlorophyll B. That is correct. In the blue region, the height of the peak of chlorophyll A is more than that of chlorophyll B. Wrong. Chlorophyll B is more than that of chlorophyll A. See, if you look at it, A and C are the same. Look at option A and option C. Both options are same. Reversing the position of chlorophyll A and B. That's all. In the red region, the peak of chlorophyll A forms at a lower wavelength than that of chlorophyll B. That is also wrong. The second one is correct. Only this blue end we will consider. Violet and indigo does not participate in photosynthesis. Towards the blue end, or we'll mention it differently. Peak. Here it is. Chlorophyll A is high, chlorophyll B is. That's all. Absorption peak. Okay. So you can eliminate this also if you want, to just keep the extremes, avoid all intervening distractions. That's all. Absorption peak, P end, blue end, red end. Chlorophyll A is low in the blue end, B is high in the blue end. Chlorophyll A is high in the red end, blue is in B is low in the red end. With reference to the, did you note down this? You have to mention the heading for the small table here. Absorption peak for photosynthetic pigments. Absorption peak for photosynthetic pigments. Spectrum, blue end, red end. So if you want to make it like the table, like this. That's all. Absorption peak for photosynthetic pigments. Absorption peak for photosynthetic pigments. With reference to the Calvin cycle, which of the given options is correct? Okay, you're still making note of it? With reference to the Calvin cycle, which of the given options is correct? So there are three questions that are asked and options are giving answers to those three questions. How many gross phosphoglyceraldehyde molecules are produced? How many gross phosphoglyceraldehyde molecules are produced? PGL molecules are produced. How many total ATP molecules are required for the synthesis of PGAL molecules? How many total NADPH2 and molecules are required for the synthesis of PGAL molecules? Phosphoglyceraldehyde molecules. Now, for every cycle of Calvin cycle, how many phosphoglyceraldehyde molecules are produced? For every cycle of Calvin cycle, how many phosphoglyceraldehyde molecules are produced? Or we will just look at it this way. Calvin cycle is for what? Calvin cycle is for carbon dioxide fixation. Correct? There are so many steps in that. Leave it. Don't bother. First is carbon fixation. Carbon fixation, reduction, regeneration. Three stages. RUBP, how many carbons? 5 carbon. RUBP is 5 carbon compound. Gets combined with carbon dioxide and gives rise to 3-phosphoglycerate. Now this 3-phosphoglycerate becomes 1,3-bisphosphoglycerate, then it becomes 3-phosphoglyceraldehyde. We will skip that one step. Now we will make it. So how many carbon it is? This is 3 carbon compound. That means for one carbon fixed, for fixing one carbon, how many phosphoglyceraldehyde, 3-phosphoglycerate is produced? 2. Correct? 2 3-phosphoglycerates are produced instead of number. Just 3 carbon, 2 in number. Correct? No ATP is used in this. Now, for 3-phosphoglycerate to become 1,3-bisphosphoglycerate, ATP is used. 3-phosphoglycerate becomes 1,3-BPG, bisphosphoglycerate. This is a 3-carbon compound, this is a 3-carbon compound. Both are 3-carbon compounds. This is also 3-carbon compound. So here, 1 ATP is used. How many times does this happen? For every carbon fixed? For every carbon that is fixed, how many times is this happening? Twice, because there are 2 3-phosphoglycerates. So, 2 3-phosphoglycerates become 2 1,3-bisphosphoglycerate. So, 2 ATP is used. Correct? Now, this 2,1,3-bisphosphoglycerate, this is also 3-carbon, becomes 2,3-phosphoglyceraldehyde, 3-PGAL, or 3-phosphoglyceraldehyde. It's also G3P, glyceraldehyde 3-phosphate. 3-phosphoglyceraldehyde is the same as glyceraldehyde 3-phosphate. How many of these are produced? 2. This is also 3-carbon. Here 2 NADPH is used. 2 NADPH is used. This is all in the process of producing, fixing one carbon. Fixing one carbon. Alright? So, RUBP to phosphoglyceraldehyde is the most important step. From here, it is all regeneration. Reduction is over. This is carboxylation, carbon fixation. These 2 steps is reduction. And beyond this, it is all regeneration of RUBP. Regeneration of RUBP. It has to be regenerated. No, RUBP has to be regenerated. Is this much clear? Now for here to here, there are so many steps, let us not bother. Regeneration. For every RUBP regeneration, one ATP is used for regeneration. 1 ATP is used for regeneration. Is this much clear? Everybody? If you look at all the steps, it is confusing. Let us not look at all the steps. Let us look at only those things which will help us understand this and answer questions. So RUBP fixes with carbon dioxide, 6 carbons. Now here you have 6 carbons because 2 of 3-phosphoglyceraldehyde is there. How many carbon has to be fixed for producing 1 glucose? 6 carbon has to be fixed. Did you make note of this? All of you write down this much. Next, balancing element. So for every carbon that is fixed, you are using 2 ATP and 2 NADPH till the production of phosphoglyceraldehyde. And you are using 1 ATP for regeneration. So how many ATPs do you use for fixing 6 carbon? How many ATPs do you use for fixing 6 carbon till phosphoglyceraldehyde? Till reduction, for 6 carbons you need 12 ATPs and 12 NADPH. For the regeneration of 6 RUBP, you need 6 ATP. This is why you need 12 plus 6 ATP, 18 ATPs and 6 NADPH. I mean 12 NADPH, 12 NADPH. Okay. Make note of this. Okay. We have to look at it from one more point of view to get sufficient understanding for answering questions. If you need expansion for this, you can write it down on the side. On the side of the paper, RUBP is ribulose bisphosphate. PG is phosphoglycerate. BPG is bisphosphoglycerate. PGAL is phosphoglyceraldehyde. Okay. See, in pathways, there will be so many steps that will be happening simultaneously. If you look at all of them together, you'll go mad. You should be able to look at it in a frame. Only the crucial, essential components of the pathways. This is carbon fixation, this is reduction, this is regeneration. Okay, done? Now I will not write number of carbons and all. We'll write it in a different way. 6 RUBP is required to fix 6 carbons, correct? Plus 6 carbon dioxide gives us, for each carbon dioxide fixed, how many phosphoglyceraldehyde do we get? 2. Gives us 12 phosphoglyceraldehyde PGL. Forget about 3 phosphoglyceraldehyde or that. Phosphoglyceraldehyde, we get 12 phosphoglyceraldehyde. So here, you make it as 10 plus 2. 10 phosphoglyceraldehyde plus 2 phosphoglyceraldehyde. Now this 2 becomes fructose 6-phosphate and then glucose 6-phosphate. Fructose 6-phosphate and this becomes glucose 6-phosphate. So we have fixed 1 glucose. How many phosphoglyceraldehyde is there now? How many phosphoglyceraldehyde is there? 10 phosphoglyceraldehyde. How many carbon atoms are there in this? Each total. Total how many carbon atoms are there? 30 carbon atoms are there. You have to regenerate 6 RUBP. Each RUBP is 5 carbon. Now for regeneration, it will regenerate 6 RUBP. Now if you look at it in carbon atoms, 6 into 5 carbon plus 6 into 1 carbon gives you 12 into 3 carbon. 12 into 3 carbon. This will give you 10 into 3 carbon plus 2 into 3 carbon. Now this 2 into 3 carbon goes to produce 1 6 carbon. 1 6 carbon. 1 into 6 carbon. That 6 carbon is glucose. Now this 10 into 3 carbon is equal to 30 carbon. This gives you 6 into 5 carbon. That's all. So for this we need, 12 ATP, 12 NADPH. Here 6 ATP. That's all. How many gross PGAL molecules are produced? With reference to the Calvin cycle, which of the given options is correct? How many gross PGAL molecules are produced? It has to be 12. But let us see if there is anything that we can Okay. See, option A and option B, option C, option D, all of them have proportion correct. For fixing 6 carbons, how many phosphoglycerol is produced? For fixing 6 carbons, how many PGL is produced? 2. For 1 PGL, how many carbon is fixed? For 1 PGL to be harnessed, how many carbon is to be fixed? For 1 PGL to be harnessed, you need to fix 3 carbons. 3 carbons. Now, for every carbon fixed, how many PGL is produced? For every carbon fixed, how many phosphoglycerol is produced? 2. For every carbon that is being fixed, 2 phosphoglycerol is produced. So if second carbon is being fixed, then it becomes 4. If third carbon is being fixed, then it becomes 6. You cannot have odd number of PGALs in the option. 3 PGAL production not possible, because for every carbon that is being fixed, 2 PGAL is produced as a byproduct then. So option 1 cannot be considered. Now option 1 and D, 4, the A and D cannot be considered because both of them are giving odd number for PGAL, whereas for every carbon fixed it is multiple of 2 that you have to consider as PGAL produced. Now we are left with B and C. 6 and 18. For 3 carbons fixed, 6 PGAL is produced, Calvin cycle. 18 is possible when 9 is fixed, but we are not looking at it. We are considering 6 carbon fixation as Calvin cycle. So 18 is also ruled out. The right choice is B, 6 phosphoglycerol. For each of that, we need 6 ATP, 6 NADPH. A more distracting option would have been 6 phosphoglycerol, 6 ATP and 9 NADPH. I mean 9 ATP and 6 NADPH. If that was the choice also, what do we choose? What do we choose? If one of the option was 6 PGAL, 9 ATP, 6 NADPH, which one do you choose? 666 or 696? Six can be considered only for partial Calvin cycle, carbon fixation and reduction. Complete Calvin cycle includes regeneration also. If 696 was also an option, what should we choose? We should choose 696. That is the right choice. But because 696 is not given, we can still consider 666. Is that clear? Excluding regeneration, there is no Calvin cycle. No? Without regeneration, how does it become a cycle? You have to start from RUBP, come back to RUBP. Only then it is a cycle. Right. Is it clear? Everybody? Now, no. It should still be 666 only. It should still be 666 only, because in the question it is how many total ATP molecules are required for the synthesis of PGAL molecules. If the question was how many ATP molecules are used, how many NADPH molecules are used, then it would have been 96. For the synthesis of PGAL, 66. Okay. After all the discussion, I forgot what the question was and I gave you 696. Okay. Next. The enzyme that catalyzes phosphorylation of the substrate without an ATP molecule is, what do you call the enzyme which can catalyze phosphorylation? What do you call the phosphorylation where the enzyme catalyzes the substrate without an ATP being produced? I mean, substrate without an ATP molecule. Okay. Some reduction should happen. Inorganic phosphate should be used for phosphorylation. but that inorganic phosphate is not coming from ATP, or the phosphate is not coming from ATP, inorganic phosphate is being used. Among the given options, which enzyme does that? Glyceraldehyde 3-phosphate dehydrogenase. If that is the enzyme, what is the reaction that it is catalyzing? Can you recall? Same reversal. Here we saw, no? 3-phosphoglycerate to 1,3-bisphosphoglycerate. Here it is saying, no, glyceraldehyde 3-phosphate is being converted to 1,3-bisphosphoglycerate in glycolysis. Reverse. If you have to choose the odd one among the given 4 enzymes, choose the odd one among the given 4 enzymes with respect to respiration. Which one do you choose? Which enzyme would you choose as the odd one among the given 4 with respect to respiration?
Option 4.
Pyruvate kinase. Why? Why do you choose pyruvate kinase? Pay attention to the question. With regard to respiration, cellular respiration, which among the 4 enzymes given below is the odd one? Option A. Why glyceraldehyde 3-phosphate dehydrogenase? Why? All the others are kinases, is it? But the question says with regard to respiration, cellular respiration. It has to be option. Option B. Why? Option B is correct. Give me the logic. Option B is correct. If option B is the odd one among the given 4 options, why? What is the logic for that? It is not an enzyme that is a part of glycolysis. The other 3 are glycolysis enzymes. The other 3 are glycolysis enzymes. Phosphofructokinase is the rate limiting enzyme of glycolysis. Remember that. How much can glycolysis happen is dependent on how much phosphofructokinase is available. Okay. Next. FAD is the electron acceptor during the oxidation of which of the following? Alpha ketogluteric acid to succinyl-CoA. What is used there? What is produced in that reaction? Alpha ketogluteric acid to succinyl-CoA. NADPH is produced. So electron acceptor is NAD+. Succinic acid to fumaric acid. What is the product there? FADH2 is the product. So the electron acceptor is FAD. That is the right choice. Succinyl-CoA to succinic acid. What is the what is happening there? GTP is being produced. Enzyme thiokinase. Okay. Fumaric acid to malic acid. Is there any energy production there? There is no energy production in fumaric acid to malic acid. Okay. All these reactions are a part of what? All these reactions given are a part of Krebs cycle. Gibberellin induces flowering in some plants only, long day plants under short day conditions, short day plants under long day conditions, day neutral plants. Does gibberellin induce flowering? Gibberellin breaks a seed dormancy, gibberellin can accelerate flowering, gibberellin can induce flowering in long day plants even under short day conditions. Okay. Cytokinins enhance longevity of the flowers. The reverse is brought about by abscisic acid inducing dormancy. brings about rapid abscission of the flower. What is the role of ethylene with respect to flower survival? Does ethylene contribute to retaining the flower, or does ethylene contribute to transforming the flower into a fruit, quickly degrade the flower? Does ethylene lead to flower senescence or flower preservation? Flower senescence. Because ethylene wants the flower to quickly become a fruit. Once it is fertilized, ethylene will be stimulated, that will lead to withering of the flower and development of the fruit. Okay, again, matching hormones with their chemical names. This is easy. Oxalis, indole acetic acid. Gibberellin is gibberellic acid. Cytokinin is zeatin. Dormin is abscisic acid. A synthetic seed consists of what? A synthetic seed consists of what is it? Somatic embryo without protecting capsule. Only sodium alginate capsule. Somatic embryo with capsule made with mercury chloride. Somatic embryo nutrient medium and capsule made with sodium alginate. What do you see? Synthetic seeds, are they produced? Do we produce synthetic seeds? Synthetic seeds are produced for rapid propagation of plants. Okay, rapid propagation of plants. You develop somatic embryos, cover them in alginate capsule, load it up with nutrient medium, and disperse it. So it is somatic embryo with nutrient medium and a capsule made of sodium alginate. Rotenone, a natural insecticide, is obtained from dairy species. What is Azadirachta indica? Azadirachta indica is neem. Bacillus thuringiensis is also used for insecticidal use. Phytophthora parvivora is a plant pathogen. It's a plant pathogen. It is not used as an insecticide. Major sources of biofertilizers. What are the biofertilizers? Bacteria, cyanobacteria, and fungi. Which fungi would you use as biofertilizer? Mycorrhiza. Mycorrhiza can be used as biofertilizer. Bacteria and cyanobacteria, nitrogen fixing bacteria and cyanobacteria are also used as biofertilizer. Which enzyme is used in the polymerase chain reaction? PCR. Restriction enzymes, reverse transcriptase, ligase, DNA polymerase. DNA polymerase. Restriction enzymes are present where? Where do you find restriction enzymes? In bacteria. Production of restriction enzymes by bacteria is a sustenance mechanism or a defense mechanism? It is a defense mechanism. Why are they producing restriction enzymes? To break down the genetic material of invading organisms. Bacteria have threat from what? What bacteria are the smallest organisms? They are the pathogens. What can cause illness in bacteria or death in bacteria? Other bacteria and bacteriophages. Bacteriophages will push their genetic material into the bacteria and try to multiply and destroy the bacteria. So bacteria have developed the mechanism to destroy their genetic material by producing restriction enzymes. Everybody wants to survive. There is one more such defense mechanism associated with all living organisms, both prokaryotes and eukaryotes. Eukaryotes presently. What is that defense mechanism? We also have it. And that defense mechanism we have or the eukaryotes have is utilized for providing insecticidal parasite resistance in tobacco. There is one parasite, a nematode. Have some mercy, we love.
Reverse transcription.
No, not reverse transcription. Reverse transcription is not an ability that is there in eukaryotes. Reverse transcription is a very special characteristic of some viruses. Retroviruses. RNA interference, gene silencing. Gene silencing. I'll explain that. I think we have a question about that. I think I saw that Melodina incognita. But we'll come there. Okay. Look at question number 106. We are moving from there. Or we'll come there. We'll come to 106 and I'll discuss that. Restriction enzymes are found in bacteria. Reverse transcriptase is found in only retroviruses, not even in all viruses. It is exclusively to retroviruses. Ligase is found in ligase. Do prokaryotes have ligase? They should have, no? They should have ligase. Prokaryotes also have ligase. Eukaryotes also have ligase joining the DNA. DNA polymerase is present in both prokaryotes and eukaryotes. E. coli cloning vector PBR322 contains restriction sites HIND3, EcoR1, BAMH1, SAL1, PUV2, TST1, CLAL, whatever. ORI, ampicillin resistance, tetracycline resistance, and ROP. What does ROP code for? What is ROP? Expand ROP. ROP in a plasmid. ROP is a short form for repressor of promoter, repressor of primer, repressor of primer, repressor of primer. ROP region is the one which determines the copy number of plasmid. ROP. ORI is origin of replication. The replication begins with ORI. ROP restricts the copy number, restricts the number of copies. Okay? ROP restricts the number of copies. You don't want the plasmid to multiply indefinitely. ROP is repressor of primer, regulates plasmid replication. Okay? Alpha-1 antitrypsin is what? It's an antacid, it's an enzyme, it is used to treat arthritis, used to treat emphysema. See, emphysema is because of the deficiency of alpha-1 antitrypsin enzyme, which will break down neutrophil elastase. Neutrophil elastase. Inflammation will lead to neutrophil infiltration. Inflammation of any tissue will lead to neutrophil infiltration. Neutrophils produce an enzyme called neutrophil elastase. Now this elastase will break down the alveolar wall, making them lose the elasticity and bloat. That is what will cause emphysema. It is a part of chronic obstructive pulmonary diseases. One of the COPD is emphysema. Alpha-1 antitrypsin is produced normally in our lungs for preventing the lungs from getting damaged, preventing the alveoli from getting damaged by the inflammation caused by anything, infection or smoke or whatever. Neutrophil elastase enzyme should be prevented. Alpha-1 antitrypsin deficiency will cause neutrophil elastase to act more on the alveoli, causing breakdown of elasticity. Treating with alpha-1 antitrypsin is a treatment to emphysema. Which nematode infects the roots of tobacco plants and causes a great reduction in yield? Melodina incognita. That is a nematode, parasitic. Truffles is mushroom, fungus. Penicillium, rhizopus, all of these are fungus. RNA interference. See, consider it this way. In lower organisms, let us say lower organisms, they have the ability to take up and absorb genetic material, DNA and RNA from the surroundings. Environmental RNA can be absorbed by them directly into their body cavity that enters the cells also. Now in this case, there is a possibility of the viral genome being in the surroundings and entering into the cells. Let's say the nematode only. We'll take the example of this nematode. This ability is there in all cells. It is there in us also. Now if a viral DNA or if a viral RNA enters into the viral double-stranded RNA enters into the cell of this nematode, what will that RNA do? The RNA will utilize the machinery of the cell itself to get integrated into the genome of the cell, multiply within the cell and destroy the cell. So what these cells do, the double-stranded RNA, whenever it is identified inside the cell, our cell also has this. There is an enzyme called dicer, D-I-C-E-R. Dicer is the enzyme which is specifically designed to identify double-stranded RNA. It can identify double-stranded RNA. When it identifies double-stranded RNA, it will cut down the RNA into pieces. Now suppose the viral genome was 100 base pairs long and you break it down into 10 base pairs of 10 pieces, 10 pieces of 10 base pairs each. If this 10 base pairs gets integrated, will it still be the same viral DNA, viral RNA? It cannot cause the disease in the organism. This is stage 1. Next stage. These broken fragments are identified by a complex, protein complex, enzyme protein complex called RISC. What is RISC? RNA induced silencing complex. RNA induced silencing complex. Now what this RISC will do? It will break the double-stranded RNA into single-stranded RNA. Break the double-stranded RNA into single-stranded RNA. Fix that single strand to itself and keep it. This is the silencing complex. Now suppose another fragment comes, you break it, you have the single-stranded RNA that is complementarily bound, destroyed. Now what is this doing? The double-stranded RNA, what will it do? For itself to multiply, it has to create a template and multiply its template. Correct? It has to get integrated into the genome of the host. It has to convert that into a mRNA. Now this mRNA will be used to build its own RNA or DNA, whatever it wants to build. What is this RISC doing? It is catching hold of a template which is complementary to whatever mRNA that can be produced, if at all this fellow has still got integrated into the genome. Are you understanding the story? Double-stranded RNA comes into the cell. Now this double-stranded RNA should get integrated into the genome of the host, trigger transcription. So this double-stranded RNA opens up within the genome of the host, mRNA is produced. So one of the strand is produced now. I am the virus. I have 2 strands of RNA that has to be produced. I get my RNA integrated into the host genome. When the transcription is happening, complementary to one of my strands, this is produced. I will use this particular component, use the other enzymes and use the enzyme that is there, build a complementary strand to that, use that double-stranded RNA, assemble the proteins, make a capsule, destroy the cell. And I go out and keep doing the same thing with others. At 3 levels, this is prevented. Now I come in, I am a 100 base pair long double-stranded RNA. I want to get integrated into the genome of the host. Before I do that, what will dicer do? Dicer will catch hold of me, cut me into pieces. Now I have a small interfering RNA, siRNA, double stranded still. I might be persistent. I might still go and get integrated. The dicer has cut me into 10 pieces. Now this RISC is catching hold of whatever pieces are available and stripping it open and keeping one strand as the template to catch hold of mRNA if at all it comes. But I am persistent. Let's say 21 to 30 base pairs gets integrated into the host. 21 to 30 base pairs get integrated into the host. Rest of the others have been taken up by RISC. Now this gets transcribed. 21 to 30 is a part of the mRNA that has been produced. Now what will RISC do? Catch hold of this mRNA and locks it up with the complementary strand to 21 to 30 that it has already built, making this mRNA ineffective. So I am not being allowed to make a copy of my genome that came into the host cell. This is the gene silencing mechanism that is there in nature. This is not exclusive to certain organisms. This is present in all eukaryotes. Now how do we utilize this for our advantage? So we are not doing anything external. We are manipulating the ability of the nematode to destroy it. So what do? We identify certain vital genes of the nematode. Vital genes, let's say the gene that is necessary for producing some survival metabolism enzymes required for metabolism, or some reproduction related genes are identified. We create double stranded RNA based on that gene. Now use TI plasmid and integrate this gene into the plant body, plant genome. Tobacco genome, we push this gene, integrate it. Now the plant genome is capable of producing, the gene of the nematode. The plant genome is capable of producing the gene of the nematode as an mRNA, as an RNA. Okay. Now when the nematode takes this, when the nematode eats this genome which is there inside the nematode is identified by the dicer. Chops off. Pieces of the genome required for producing a gene essential for reproduction is integrated into the RISC. Now when this gene is transcribed, the gene of the nematode which is required for the nematode to reproduce is transcribed and mRNA is produced, RISC will catch hold of it and destroy that mRNA, not allowing that particular gene in the nematode to express. Now if you are not allowing the gene which is required by the nematode to survive or to reproduce to express, the nematode will eventually die. It is not like chemicals which will die the insect instantly. The insect which is trying to attack this will not be able to reproduce. What do these parasites do? They get into the roots of the tobacco plant. They survive there. They multiply and all that. If they are multiplying, the root is destroyed. But a nematode infects, it starts eating the plant. The interfering gene gets into the nematode. The nematode is not able to survive, or it is not able to reproduce, thereby protecting the host who you are supposed to protect, tobacco in this case. A genetically engineered microorganism used successfully in the bioremediation of oil spills is a species of which is used Pseudomonas cupida. Pseudomonas. Bioremediation is using organisms, living organisms or biological mediators for clearing harmful substances from the environment. Which bond forms an ester bond in nucleotides? Ester bond in nucleotides, sugar and phosphate, sugar and base and phosphate, nucleotides of opposite strands. Sugar and phosphate. Bond between sugar and base is glycosidic bond. Is there bond formed between base and phosphate? No bond between base and phosphate. Nucleic acids can be fragmented by nucleases. It's a easy question. An mRNA chain has 66 nitrogen bases. The last 3 bases are UAG. What will be the number of functional codons and amino acids in the polynucleotide chain? How many codons are there in this chain? How many codons are there if there are 66 amino acids? 22 codons. How many amino acids would this polynucleotide produce? If there are 22 codons in this, how many amino acids would it produce? 21. Why? The last codon is UAG, which is a stop codon. Give me the names for the stop codon. Names for the stop codon, not the code. Opal, ochre, amber. UAA, UAG, UAC. Nowashchin discovered a triple fusion in which plant? Lilium and Ventilaria. What? Given no. You have written in your book. Okay. Identify the incorrect match among the following. What is used for what? Arachis is groundnut, oil and fodder. Is it correct? Is it used for oil and fodder? Groundnut cakes, groundnut oil cakes are very rich sources of protein for cattle. It is used for cattle feed, as cattle feed. You know, you get this soya chunks, that dry ball like soya. What is it? It is oil cake. Earlier, these were all used as cattle feed. Now humans are feeding on it. Even millets were used as cattle feed earlier. People who would eat millets were considered to be poor people. Earlier times, people who would eat polished rice, white rice, were considered to be rich people. In our village and all, people used to say they eat white rice. Okay, village people would not eat much. Even now, if you ask old grandmothers, 80, 90-year-old grandmothers, they would say we would not eat these things. And even the oil cakes are the cattle feed. You get it in cages. What is oil cakes? When these seeds are pressed and oil is extracted completely from that, whatever dry husk is left out, that is pressed and made into cakes. Oil cake. You wet it, it all loosens up, and the cattle will eat it. High source of protein. Soya bean is pressed and oil is extracted. When complete oil is extracted from soya bean, whatever husk is left out is very high source of protein, and that is pressed, dried, and cut into small pieces and chunks, and sold as those soya chunks which you eat. Very economical. It is about 140-150 rupees per kg. 50 grams per 100 grams. 50% is the protein content. 50%. No oil in that. And it is tasteless. You can add it to anything. High source of protein. Meat, red meat. How much is the percentage protein availability in red meat? Do you know the protein concentration in different items? Pulses, 18 to 20%. 18 to 20%, all pulses, mostly. Soya bean is the highest among plant seeds. Soya bean has about 26 to 28% protein. Red meat is about 30% protein. Red meat is about 30% protein only. One egg, 6 grams protein. One egg is 6 grams protein. 100 ml of milk gives you 3 grams protein. Soya chunks give you 50% protein. Every 100 grams give you 50 grams of protein. So look at the cost implication. 1 kg of red meat. How much is it? Around 300? How much is 1 kg of red meat? 600, huh? 600. Okay. So for 600 rupees, 30% if you consider, you get 300 grams protein. For 600 rupees, you get 300 grams protein. Digestibility of red meat is low. Our system cannot digest it completely, and the bioavailability is low. Absorption is lesser. So digestibility and bioavailability of plant proteins is higher than animal proteins. So consider red meat versus soya chunks. Alright, 150 grams. 150 rupees you get 1 kg of soya chunks, which will give you 500 grams of protein. So per gram of protein, how much are you spending? Per gram of protein you are spending 60 paise. You are spending 60 paise for in meat. Per gram you are spending 60 paise. Correct, no? Per gram of protein, available protein, soya chunks, how much are you spending? How much are you spending? For 150 rupees you get 500 grams. So 150 divided by 500. 150 divided by 500. No. 500 divided by 150, no? For 150 rupees you get 500 grams. 500 grams, 150 rupees. 1 gram, how much? Cross multiplication. 500 divided by 150. 500 divided by 150. About 33 paise. About 33 paise. For 1 gram of protein, you are spending 30 paise in with soya chunks. For 1 gram of protein, you are spending 60 paise in red meat. Double. No, more than double. It is 1/4. In 600 rupees, you get 4 kgs of soya chunks. 4 kgs means you have 2000 grams. 3 times more. 3.5 times more. That is 1/3. 1/3 is the cost. 1/3. More environment friendly, more protein rich. If you want to build muscles, bodybuilders use soya chunk. Okay, total area, fiber and fodder, total area, sun hemp, fiber and fodder is correct. Trigonella, leaf vegetable and medicinal. Trigonella, fenugreek, methi. Trigonella is methi. Is it used for leaf vegetable and as medicinal medicinal plant? Yes. Pterocarpus, fodder and green manure. Pterocarpus is sandalwood. It is not used as fodder and manure. It is much more expensive than that. Taxonomists to popularize the binomial system. Who is the one? Binomial system. Linnaeus. Scientists and their contribution. Joseph Priestley, Hanning and Hutchinson, Benson and Basham. Okay, which one are you confident about? Joseph Priestley, apparently exchange of oxygen and carbon dioxide. I don't think these are given in NCERT, no? Are these fellows present in NCERT? They are present in NCERT, unfortunately. So you have to remember their contribution. Joseph Priestley, exchange of oxygen and carbon dioxide. He is the one who discovered oxygen is synthesized during photosynthesis. Hanning and Hutchinson, tissue culture, plant tissue culture. Benson and Basham, plant physiology. Calvin cycle, he is the one who contributed to, they are the ones who contributed to Calvin cycle discovery. Which pairs show the same morphology of modified structure? Lathyrus, tendrils. Lathyrus is sweet pea. Tendrils are produced, yes. Smilax also produces tendrils. Ziziphus and Argibone both produces spines. Now, how do we say which one is what? Ziziphus is modified stipule, argemone is modified leaf, though both of them produce pine. So it is 1 and 4, Lathyrus and Smilax are tendrils, both are modified leaves. It is justified that potato tuber is an underground stem because, how can we justify potato tuber to be an underground stem? What does it have? It has axillary buds. Storage, reserve food is not a criteria to determine whether it is root or stem. Phyloclades are what? Modified stems found in xerophytes, found in all xerophytes, modified assimilating stems bearing flowers only found in xerophytes. Phyloclades are flat leaf-like photosynthetic structures with storage. Store water and help the plant survive in dry conditions. So phyloclades are found in xerophytes. They are modified stem. But are they found only in xerophytes? They are not found only in xerophytes. They can be present in other environments also. And all xerophytes do not have phyloclades. There is thorn modifications also. Vascular bundle characteristics with plant parts. Medullary rays, pericycle with dedifferentiating ability, lignified hypodermis, mesophyll with heterogeneous chlorenchyma. Which one are you sure about? Medullary rays, pericycle with dedifferentiating ability, lignified hypodermis, mesophyll with heterogeneous chlorenchyma. Where do you find pericycle? Pericycle is found in dicot roots. Pericycle is found in dicot roots. And pericycle is the structure from which what originates? What comes from the pericycle? In roots. Lateral roots. Lateral roots grow from pericycle. That is why the dedifferentiating ability is significant there. Lignified hypodermis is found in. Lignified hypodermis is what makes your hand get cut when you try to pull on this. Monocot leaves, grasses. If you try to pull on the grasses, your hands will get cut because of that lignification. Mesophyll with heterogeneous chlorenchyma. Mesophyll with heterogeneous chlorenchyma. What is heterogeneous chlorenchyma? Palisade parenchyma and spongy parenchyma. Okay. It is in dicot leaves. Dicot leaves. Medullary rays are a characteristic of dicot stem. Identify the incorrect statement regarding vascular bundles in monocot stem. All vascular bundles in ground tissue are scattered irregularly. Is it correct? Irregular scattering of vascular bundles in monocot stem. Do we have this anatomy? We have this, no? We still have this. In a vascular bundle, xylem and phloem are separated by cambium. Is it right? Is it right? In monocots? Monocots have closed vascular bundles. Closed vascular bundles mean there is absence of cambium. There is no secondary growth. So option statement B is wrong statement. That is why that is the correct choice. The others are also correct. Y-shaped arrangement of xylem strands, vascular bundle covered by a bundle sheath. Next one, heartwood, sapwood, rolled out. Okay. Vein ending epitheme cavity and water stoma are part of stomata, hydathode, osmophores, nematode. Where do we find this? In hydathode. Even if you have not learned this, hydathode is the ending of veins. That you should know. Epitheme cavity is where the water is stored before it comes out. What are osmophores? Osmophores. Osmophores are the fragrance releasing structures in plants. They are the ones which release fragrance, attracting insects during pollination. Stomata, you know, Nematode, nematodes, nematophores. Where do you find nematophores? Nematode are a part of nematophores. Mangroves. Mangroves. What are these nematophores? Breathing roots. Nematophores grow against gravity. So what tropism does it show? Negative geotropism. Negative geotropism. Identify the incorrect statement regarding tylosis. What are tyloses? Tyloses are balloon-like outgrowth from the stem coming from the xylem parenchyma. Balloon-like outgrowth from the stem coming from the balloon parenchyma. From the parenchyma, xylem parenchyma. Incorrect statement. Obstruct conduction of water in old and injured vessels. Is it right or wrong about tylosis? That is one of the reasons why you are producing tylosis, to prevent water from flowing in old and injured vessels, so that the efficiency of water transport is increased. Balloon-shaped structures formed from xylem parenchyma, that is what they are. Check the spreading of pathogenic fungi. Yes, by blocking the movement of this fungi. Initially thick-walled, later disappear. No. They are not thick-walled, they remain permanently. The two chromatids of a metaphase chromosome represent what? Replicated chromosomes to be separated at anaphase, homologous chromosomes of a diploid set, non-homologous chromosomes joined at the centromere, maternal and paternal chromosomes joined at the centromere. What do the chromatids of a metaphase chromosome represent? Are they maternal and paternal chromosomes joined together? No. They are duplicate copies of each other, replicated chromosomes which will be eventually separated during anaphase. If it was the same set of options and the question was the two chromosomes of a metaphase 1 of meiosis represent. Two chromosomes of a pair of metaphase 1 represent what? Same options. Replicated chromosomes to be separated at anaphase, homologous chromosomes of a diploid set, non-homologous chromosomes joined at the centromere, maternal and paternal chromosomes joined at the centromere. Option B. Option B. Can you make option D also right? Can you make option D also right? For the same question, meiosis 1, metaphase, pair of chromosomes, you have to make option D right option. What will you change in option D to make it right? Change something in option D, make it right. What will you change? Come on, quickly. How do you make it right? Can we consider the homologous pair to be maternal and paternal chromosome pair? If the option D was a homologous pair of chromosomes representing maternal and paternal chromosome, why not? How did you get the homologous pair? The homologous pair, one of the pair comes from the mother and one comes from the father. No, that is how the set is made. Homologous pair. Let's say chromosome number 1. Where did you get both of these chromosomes from? One from the mother, one from the father. So, can we not consider homologous pair as one of them coming from the mother and the other coming from the father? It is no. So that also can be made right by modifying the statement. Then suppose I have modified that statement D. Now which one do you choose? B or D? Homologous chromosomes of a diploid set representing maternal and paternal chromosomes is the option D. Which one do you choose? B or D now? Which one will be appropriate? B is homologous pair of a diploid set. D is that plus an extended explanation telling that it represents maternal and paternal set. What do you choose? D would be more appropriate then, because it is going into more detail. Next question. Daughter cells of meiosis 1 receive half the number of chromosomes as their parent cell, have the same chromosome number as the parent, contain double the chromosome number compared to meiosis 2 daughter cells, receive half the number of chromosomes due to disjunction. What is right for daughter cells of meiosis 1? Come on, you should speak. So much silence is not good. What is right about the daughter cells of meiosis 1? Between 1 and 2, is it? Statement 1 and 2? What? So what will be the number of chromosomes in the daughter cells of meiosis 1? Will it be half or the same? Half. The number of chromosomes reduced in meiosis 1, DNA content reduced in meiosis 2. Correct, no? If it was a combination of statements to be selected as options, and one of the statements would be have the same amount of DNA as that of the parent, wouldn't that also be right? At the end of meiosis 1, daughter cells have half the number of chromosomes and the same amount of DNA as that of the parent. Yes or no? Half the number of chromosomes, but the same amount of DNA. Meiosis 2 is non-reductional with respect to the number of chromosomes, but meiosis 2 is reductional in terms of the content of DNA. Meiosis 1 is non-reductional in terms of the amount of DNA, but it is reductional in terms of the number of chromosomes. So, the references you get here and there about meiosis 2 being like mitosis is wrong. Alright? Being like mitosis, what is the same? There are two components. I can say meiosis 1 is like mitosis in terms of reduction in the content of DNA. Can I not say that? Because in meiosis 1, between the parent and the daughter cell, there is no reduction in the content of DNA. Same as mitosis, there is no reduction in the content of DNA, in the content of DNA between the parent and daughter cell in mitosis. So that statement comparing or equalizing meiosis, part of meiosis with mitosis is wrong. Dichogamy favors cross-pollination because what is dichogamy? Differential maturation of the reproductive whorls. Androecium and gynoecium mature at different times so that it cannot do autogamy. Autogamy is not possible. When the stamen is mature, if the carpel is not mature, stamen cannot pollinate. Likewise, vice versa. Dichogamy favors cross-pollination because anthers and stigmas are placed at different levels, no stamens and stigma mature at different times. That is the right choice. Which of the following diseases are caused by bacteria? Now, look at the options. Try to identify viral diseases. If any option has a viral disease, you have to eliminate that. So option A, AIDS is a viral disease, leave it. Option B, typhoid, tuberculosis, tetanus are all bacterial, but pneumonia can be viral also. Pneumonia can be viral also. So B is, keep it aside, we will see if there is any unambiguous option. Option C, polio, hepatitis, scurvy, beriberi, leprosy. Polio and hepatitis are viral. Measles, mumps, malaria, sleeping sickness, syphilis. Measles, virus. So option B. In that case, pneumonia is to be considered as bacterial pneumonia. If there was another option, let's say typhoid, tuberculosis, gas gangrene, tetanus, what would you choose? Would you choose the option with pneumonia or the one with gas gangrene? Which one do you choose? Don't know what gas gangrene is. I have told you, Clostridium perfringens. Clostridium perfringens causes gas gangrene. It produces toxic gas which will separate the muscle from its fascia. Gas crawls around under the skin, leading to gangrene of the affected parts. Crepitations can be heard if you just squeeze the limb or whichever part that has been affected by gas gangrene. You will hear crepitations. What are crepitations? Like you crush dry leaves in your palm. You get that sound like air escaping from space. Okay. Can you tell me the causative agent for cause for each of the diseases mentioned here? AIDS, quickly. HIV, human immunodeficiency virus. Hydrophobia, it's not a disease, it is a sign of rabies, caused by rabies virus. Pneumonia, most common pneumonia is streptococcal pneumonia, streptococcus. Influenza also can cause pneumonia, Haemophilus influenza, viral pneumonia. Okay. There can be aspiration pneumonia also. What is aspiration pneumonia? One of the major complications of corrosive swallowing. People attempt suicide by consuming some corrosives, kerosene, all that nonsense, those toilet cleaners, such things. It leads to irritation of the stomach and it will cause vomiting. And when vomiting happens, when violent vomiting happens, there is a possibility of that vomitus entering into the airway. And depending on what is entering the airway, it can lead to irritation of the airway and can cause pneumonia. That is called aspiration pneumonia. Aspiration pneumonia. Alright? What is pneumonia? Pneumonia. See, in the respiratory passage, as you go down, inflammation of the pharynx, pharyngitis. Inflammation of the larynx, laryngitis. Trachea, tracheitis. Bronchi, bronchitis. Bronchioles, what do you say? Ruled out, ECD. Okay. Rhodophyceae dictyota. May not be sure about it. It's an organism. Let it be. Chlorophyceae non-motile gametes. Is it right? Chlorophyceae, green algae, they also do have motile gametes. So anything without C can also be ruled out. So option D can be ruled out. B and D ruled out.
Yes.
A and C is the no. A is also ruled out because D, Rhodophyceae are phycoerythrin. Phycoerythrin is the pigment that you find in what? rhodophysian, red algae. So the right choice is A and D. Keystone species deserve protection because, what are keystone species? Critical species which are required for the maintenance of the ecosystem. Those are keystone species. Removing them will lead to ecological imbalance. So, keystone species deserve protection because they play an important role in supporting other species. Tropical dense forest exists due to, this is an easy question. What will lead to tropical dense forest? The conditions which are required for plants to grow, water and temperature, high rainfall and high temperature. High rainfall, low temperature is characteristic of what kind of forest? Temperate forest. Rainfall is good, but temperature is low. Low rainfall, high temperature, deserts. Low rainfall, low temperature, cold deserts, tundra. Next. In the vast marine ecosystem, some seas develop a red coloration due to the presence of. Red coloration in oceans is called what? Red tide. Red tide is caused because of dinoflagellates. Name them. Goniolax. Goniolax. Is it good? Is it good for tourism? Red ocean. All reel makers will throng the place. Go there and make a reel with Red Sea in the background. Horrible reels people are making nowadays. Some fellow has taken his Mahindra Thar on the railway track. Okay. And when the train starts approaching, he wants to show the power of Thar and wants to take it out in reverse. Fortunately, within time he got out. Any guesses where he is right now? Jail. Not hospital. On the track. But because of his perversion being off track, he is in jail. Stomatal apparatus identified the correctly labeled parts. Epidermal cell, subsidiary cell, stomatal aperture, guard cell. A is epidermal cell, B is subsidiary cell, C is stomatal aperture, D is guard cell. Guard cell shape in monocot and dicot. What is the shape of guard cell in monocots? Dumbbell shaped. In dicots it is bean shaped, kidney shaped. Ground tissue includes what? There are three types of tissues in plants: epidermal tissue, ground tissue, and vascular tissue. Vascular tissue is xylem and phloem. Epidermal tissue is epidermis and all its appendages. Everything else is ground tissue. So cortex is what? Cortex is ground tissue. Endodermis is ground tissue. Pericycle, ground tissue. Pith, ground tissue. Medullary race, ground tissue. Except the vascular bundles, which are vascular tissue, and the epidermis, which is epidermal tissue, everything else is ground tissue. So all tissues except epidermis and vascular bundles. We'll take a 5-minute break. Okay, quickly. If somebody wants to take a break, 5-minute break. Then when we come back, we'll do the zoology. Snails don't have tentacles, they have muscular foot. Chiton is a mollusk, creeping foot. Germ cells of sponges are, if we talk about germ layers, sponges are sponges diploblastic, triploblastic, what are they? The body of the sponges is derived from ectodermal and endodermal tissue. There is no mesodermal tissue in sponges. Though we cannot call sponges technically as diploblastic also, but you can consider both germ layers. So germ cells are derived from both ectoderm and endoderm. Both ectoderm and endoderm. Spermatocytes are of endodermal origin, oocytes are of ectodermal origin. What is that special cell that you find in sponges called? Choanocytes or collar cells. Choanocytes or collar cells. Post-anal tail is present in chordates, vertebrates, invertebrates, all of these. Post-anal tail is a characteristic feature of chordates. There are 4 characteristic features of chordates. What are the 4? 4 defining characteristic features of chordates. What are they? The presence of notochord, dorsal, hollow, nerve, root, post-anal tail and pharyngeal gill slits. These are the 4 essential characters to be identified as chordates. Which of the following is not a cell of areolar tissue? Areolar tissue is loose connective tissue. Areolar tissue is loose connective tissue. Which is the densest connective tissue that you can find? Dense connective tissue. Tendons and ligaments. Which of the following is not a part of areolar tissue? Plasma cells, adipose cells, macrophages, Schwann cells. Schwann cells. Where do you find Schwann cells? Schwann cells are part of the nervous system. In the nervous system, see, Schwann cells are not a part of this areolar tissue. For any connective tissue, you need to have what is necessary for connective tissue? You need to have a ground substance and you need to have cells in the ground substance. Okay. Nervous system has 2 types of cells. Those which are supposed to perform the neural activity, that is the neurons, and those which perform the supportive function. What are they called? Supportive cells for the nervous system are called neuroglial cells. Schwann cells are one of the neuroglial cells. Alright? There are different types of glial cells you can make note of. There are neuroglial cells in both central nervous system and peripheral nervous system. Alright? Astrocytes are the cells which will form the blood-brain barrier. Astrocytes. They form the blood-brain barrier. Important. You should not allow anything that is in the blood to cross this and enter the nervous system. There is regulation, extra level of protection in the blood vessels that enter the nervous system. That is the blood-brain barrier. Correct? Astrocytes, blood-brain barrier. Oligodendrocytes. Oligodendrocytes and Schwann cells have similar function. What is the function? The function of oligodendrocytes and Schwann cells is the same, but the location of action is different. What is the function and where do they act? Both oligodendrocytes and Schwann cells produce myelin sheath. Oligodendrocytes and Schwann cells produce myelin sheath. Oligodendrocytes produce myelin sheath around the axons of the central nervous system. Schwann cells produce myelin sheath around the axons of the peripheral nervous system. Peripheral nervous system. Okay. Then you have microglia. Microglial cells are the defense cells, immune cells. Ependymal cells. Ependymal cells are a part of the ventricular system. Ependymal cells produce cerebrospinal fluid. Ependymal cells produce cerebrospinal fluid. Then that's all. These are the important cells. Astrocytes, microglia, ependymal cells, oligodendrocytes, Schwann cells. Oligodendrocytes and Schwann cells are for myelin sheath. Ependymal cells produce cerebrospinal fluid. Astrocytes form the blood-brain barrier. Microglia are the defense cells. 5 cells, you remember. 5 types of glial cells. Which cells have the shape of signet ring? Signet ring. How does the signet ring appear? Which of the cells mentioned have the shape of the signet ring? Like this. Which of the given cells have it? Mast cells, osteocytes, adipocytes, melanocytes. Adipocytes, fat cells. What is this in the adipocytes then? Nucleus. What is this? Fat droplet. Fat droplet. See, adipocytes are stretchable. They can expand. The cells, they will resist expansion initially. Initially, adipocytes resist expansion. Have you noticed some people who would be, they would have told, you would not have experienced in your life so yet, still young. They would be very thin for a certain period of time, very lean. They don't put on weight. So, considering that as a permanent advantage, they will continue to eat. And at some point of time, their body starts putting on weight. And then once they put on weight, they will not be able to get back to a lean shape. Adipocytes will resist expansion initially. They will try to metabolize. There is mechanism to metabolize and all that. There is brown fat, brown adipose tissue in the neck pad and all that. Brown adipose tissue will increase metabolism and try to keep the accumulation low. But once expansion happens, you expand the adipose cells, they expand and keep on accumulating fat. Even if you lose that, their tendency to expand has increased. Anything available to accumulate, they will accumulate and expand. That is why people lose weight over a long period of time and regain weight very quickly. To lose is long, it takes long. To put back, 1 month, 2 months. Okay. Which of the following amino acids has a hydroxymethyl group as its functional group? Serine, proline, alanine, arginine. Hydroxyl group is serine, threonine and tyrosine. You have to remember sulfur containing amino acids, cysteine and methionine. That is a frequent question. Cysteine and methionine, CM, cysteine, methionine, sulfur. Serine is hydroxy group. Questions are more likely to explore acidic and basic amino acids. Acidic amino acids are only 2 out of the 20 that our body has. What are the 2 acidic amino acids? Glutamic acid and aspartic acid. There are 3 basic amino acids. The rest of the 15 are neutral. What are the 3 basic amino acids? HAL, histidine, arginine, lysine. HAL, histidine, arginine, lysine. Next, pick out the wrong statement. Amino acids are substituted methanes. Is it right or wrong? Amino acids are substituted methanes. CH4 is methane. Amino acids have one functional group, one amino group, one carboxyl group and one hydrogen. So amino acids are substituted methanes. Glycerol is trihydroxypropane. That is also correct. Lysine is a neutral amino acid. Is it right? Lysine is a basic amino acid. Lecithin is a phospholipid. That is correct. During an injury, the nasal septum gets damaged. Nasal septum. Nasal septum is the partition between the 2 nostrils. That is the nasal septum, not this. This is tip of the nose. For recovery, which cartilage is responsible? So nasal septum is made up of what cartilage? Nasal septum is made up of hyaline cartilage. Recovery also is by production of more hyaline cartilage. Okay, next 2 questions. Digestion. We have to let it go. But tell me, which of the following are absorbed in the alimentary canal without digestion as such? Albumin, of polysaccharides, fat-soluble vitamins, proteins. Which does not require digestion among the four? It will just absorb, it is absorbed as it is without any modification. Vitamins. Vitamins are absorbed just like that. Vitamins and minerals, they just have to be released from their binding with the other macronutrients. Micronutrients will just get absorbed. Enzymes and source will leave it. Respiratory center in the brain is stimulated by. See, we know carbon dioxide stimulates respiratory center, not oxygen level. Carbon dioxide in the artery or the vein. Carbon dioxide present where is the one that stimulates respiratory center? Is it the venous concentrated level of carbon dioxide or the arterial carbon dioxide? Huh? Artery. Veins will have higher level of carbon dioxide only. But if the arterial blood has higher level of carbon dioxide, then there is a problem. Who senses that? Yesterday we saw carotid body and diuretic body. They are chemoreceptors. Carotid body and diuretic body are chemoreceptors. Because, see, they are present here only, in the arch of aorta and in the carotid arteries. So carotid arteries and arch of aorta are very close to the left ventricle. That means that is the closest you can get to the oxygenated blood source. Oxygenated blood source is left ventricle. From there the blood is coming out, arch of aorta and this. Chemoreceptors are positioned there. So that you can pick up the carbon dioxide concentration very easily and more accurately. Where are these centers? Where are these respiratory centers? Where is the respiratory center? In the medulla. Respiration is controlled by cerebellum, medulla oblongata, olfactory lobes, hypothalamus. Medulla oblongata. Olfactory lobes are for what? Olfaction. The nerve endings. See, olfactory lobes are present adjacent to a perforated plate of bone at the base of the skull. Perforated plate of bone at the base of the skull. And the nerve endings of the olfactory nerve are exposed and open at the base, at the roof of the nose. Nerve endings are open, they are open nerve endings at the roof of the nose. Alright? So when you sniff, what are you doing? To get the smell of something, what do? You will sniff. When you sniff, what are you doing? You are pushing air higher up in the nose. You are pushing air higher up in the nose to reach the roof of the nose, where the nerve endings are just hanging. It's called cribriform plate. Cribriform plate. The bone is called cribriform plate. Nerve endings are perforated passing through that. And when the chemicals which are causing the odor is reaching this nerve ending, that will trigger smell. Have you experienced this? When there are fumes, toxic fumes, some toxic fumes are there, and you inhale the toxic fumes, suddenly you will feel a kind of, instantly you will develop that headache and kind of irritation at the top of the nose. Inside the nose you will feel some burning sensation and irritation. That is because of the triggering of the nerve endings of the olfactory nerve, which is very sensitive. Okay. During normal inhalation also, if the chemicals producing odor are high in concentration, it will reach the nerve ending. Otherwise, you have to sniff. Right. Especially when you are sniffing for gas leakage, LPG leakage. Somebody tells, I smell LPG. Is the gas connection proper? You go and sniff around the cylinder, around the gas stove. And associated with this olfactory nerve only in the same area, 13th nerve is also identified apparently. Isolation is not yet done. 13th nerve or the 0th nerve or the nervus terminalis, 13th cranial nerve. What is the function? I told you yesterday. Detection of pheromones. Why not olfactory nerve only? Olfactory nerve is supposed to evoke survival response. This 13th nerve or 0th nerve or the nervus terminalis is apparently attributed to the function of in enhancing the reproductive possibility. Sensing pheromones is an opportunity for reproduction. So keeping it separate. Human body and the functioning of human body is said to be like the ocean. We know about how human body functions as much as we know about how ocean operates. What is there in the ocean, and there is as much about the human body left to discover as there is about the ocean left to discover. In both cases we know so much already. We know so much about the ocean, oceanology, and you have multiple masters, multiple specialization within the ocean studies only. Same with human body also. There is so much that is already known. But what is known is tremendously minute compared to what is unknown. That's the fun. Okay. And people who are experimental evolutionists, they are the spiritual people. They experiment with their own existence and try to explore the possibilities or they explore the limits of their own evolution. They say that it is not something that can be done en masse. The understanding of human beings beyond what our intellectual faculties allow is a matter of realization and experience. So going deep into understanding oneself is the only way to cross the boundaries of this limited existence. I don't know how much you understood, but it is very profound. You are truly infinite. Infinity is life. That's all. Every self is infinitely mysterious. So that's the beauty of life. That is why you should refrain from passing conclusions about life. This is all it is. That's all. This is all it is. So far, to the extent of my understanding, this is what I know. So, relativity is important there. 148. Each stroke volume of 70 ml of blood is pumped by each ventricle. Now, stroke volume is the volume of blood pumped by each ventricle or both ventricles put together. Each ventricle. Cardiac output is the volume of blood pumped by each ventricle or both the entire heart put together. Each ventricle. Cardiac output is stroke volume into heart rate. Stroke volume into heart rate. That is the basic understanding we need to have for this question. Each stroke volume of 70 ml of blood is pumped by each ventricle. That is a true statement. The duration of a cardiac cycle is directly proportional to the number of heartbeats. Is it true? Duration of the cardiac cycle will be more if the number of heartbeats are less. If heart is beating faster, the cardiac cycle becomes shorter. It will be completing faster. So it is inversely related. Statement 1 is correct and statement 2 is wrong. Assertion is true, reasoning is false. When the reasoning is false, the explanation doesn't matter. Cardiac pacemaker in the patient fails. Where will the artificial pacemaker be implanted? Sinoatrial node. But there is a problem to this question. Sinoatrial node is correct. Pacemaker is implanted. The pacemaker is what? It is an artificial electricity producing device. It can be tuned, it can be set to produce certain impulses, certain number of impulses per minute. The intensity of the impulse, the frequency of the impulse, all of that can be set in the pacemaker. Now, where should this impulse reach? Where should this impulse reach? It should reach the heart. It should reach the heart. Now, suppose sinoatrial node has failed. SA node has failed. There is failure of sinoatrial node. Impulse should reach where now? Impulse from the pacemaker should reach where? Sinoatrial node has lost the ability to generate an impulse. Now the pacemaker has been implanted. Connection from the pacemaker has to reach the atrial bundle of the sinoatrial node. So at the junction of the sinoatrial node and the atrial bundles. And also the atrioventricular node, AV node, correct? So that the atrium is also stimulated and AV node is also stimulated. Both the atrium and the AV node are getting impulse at the same time, like they would have got from the sinoatrial node. So there is a pacemaker and there are electrodes. There has to be wires, electrodes that will reach the particular location. Now if AV node is also failed, SA node is failed, AV node is also weak, then from the pacemaker, now this becomes biphasic. Single phase pacemakers are there, double phase pacemakers are there. Now single phase pacemakers are like SA node. Pacemaker, SA node, AV node. Now if SA node failed, SA node has failed. From the pacemaker, this is single phase, phase 1, phase 2. Let us consider these are the two phases. Phase 1, phase 2. Single phase goes to the SA node. So this is the atrial fibers. And the same from this only goes to the AV node. AV node is normal, AV node is functioning. So single impulse from this goes to the SA node, supplies the atrium. Same time it has gone to the AV node also. AV node delays and fires. AV node is functioning. But if AV node also has become weak or AV node also has failed, then what happens? From this phase 1, it goes to the SA node and the atrial fibers. From phase 2, it goes to the AV node, and there is an internal connection between these. The delay is set. You will set the delay. Phase 1 fires. Whatever is the delay that you have set in the pacemaker, after that much delay, the phase 2 fires, biphasic pacemakers. You understand? It's just generator with supply. Such micro level. Enhances the ability of people to survive. Otherwise they would die. So pacemaker is implanted under the clavicle here. Under the clavicle, and the electrodes are transferred to the heart through the arteries. Through the artery, reverse, like angiogram you do, you pass the electrodes to the respective location through the arteries or vessels and connect the electrodes to the respective places. Like that. In this case, we will just consider it as sinoatrial node, but you should know the mechanism. Pacemaker is not put into the heart. Pacemaker is external. Because anytime there is a malfunctioning pacemaker, you have to repair it. It's an attachment. Next, you are required to draw blood from a patient and keep it in the test tube for blood analysis. Which test tube should not be used? Test tube containing calcium bicarbonate, chilled test tube containing heparin, test tube containing sodium oxalate. What do you not want to use? Test tube with calcium bicarbonate should not be used because calcium promotes clotting. Calcium promotes clotting. So clotting should not happen. Heparin is an anticoagulant. Chilled test tube is good. It will prevent enzymatic degradation of RBC blood cells. Oxalates, sodium oxalate. Oxalate will bind calcium and prevent clotting. You have to remove calcium from the blood sample. How do you remove? Oxalate. Oxalate is the anticoagulant. EDTA is also used. EDTA. EDTA and oxalate are used for coagulation. Anticoagulation. Yellow color of urine is due to urochromogen, urochrome, carotene, none of this. Urochrome. Where is it coming from? What is the source of urochrome? What is the source of urochrome? Okay, just quickly know this. In the blood, RBC destroyed, RBC dies, what will it release? Heme plus globulin, recycle. Globulin is a protein, it gets recycled. That will again be broken down and used for, the amino acids are used. Heme gets converted into biliverdin. Biliverdin becomes bilirubin. This is unconjugated. This is unconjugated. Bilirubin plus albumin. Albumin is the escort. Albumin is the escort for bilirubin. Albumin will catch hold of bilirubin and takes it to, where does it take it? Where does albumin carry bilirubin to? It takes it to liver, delivers it in the liver. This is in the blood. Now here, liver. This enters the liver. Now bilirubin is here, albumin exits. Right? Now this bilirubin will get combined with glucuronate. Bilirubin glucuronate, that is conjugated bilirubin. Conjugated bilirubin. Conjugated bilirubin will go into, what is this? Gallbladder. Alright. Conjugated bilirubin goes into gallbladder. From gallbladder, it goes into intestine. Bilirubin goes into the gallbladder, stored in the gallbladder, excreted into the intestine. In the intestine, bilirubin gets converted into stercobilin. Stercobilin is excreted in the feces. Stools get yellow color because of stercobilin. Bile. If there is liver problem, stercobilin will not be there. If there is obstructive jaundice, that is, there is some gallstone, it is preventing the bile from coming into the intestine. Bilirubin cannot become stercobilin. Stercobilin does two things. It reduces the odor in the feces. Kind of, it's a deodorant for the feces. Alright, stercobilin is deodorant and colorant. It will add color and it deodorizes stools. So people with obstructive jaundice will have dark colored stools. This is trying to conjugate as much as possible. So this is normal. There is no problem with this fellow. Whatever unconjugated bilirubin is coming, he is trying to conjugate it. But the problem is, there is too much of bilirubin being produced. Why? It can be because of too much of RBC destruction. You understand? This is the sequence. So, bilirubin is coming from heme. Did you understand the story? That's all. So, when you look at the blood report, bilirubin, total bilirubin conjugated bilirubin and unconjugated bilirubin. You can find out where the problem is. Total bilirubin is more. That means something is happening. There is more bilirubin. Of that, conjugated bilirubin level is normal or slightly raised, but unconjugated bilirubin level is very high. What kind of jaundice is this? Pre-hepatic, before the liver, hemolytic, breakdown of RBC. Conjugated bilirubin is low. Conjugated bilirubin is low, unconjugated bilirubin is high. Then, conjugated bilirubin is low, unconjugated bilirubin is high. Then what does it mean? Conjugation is not happening. Hepatic jaundice, hepatitis, inflammation. Unconjugated bilirubin is normal only or slightly raised. But conjugated bilirubin level is very high. Meaning what? Conjugated bilirubin is not getting excreted. Probably obstruction, gallstone, obstructive jaundice. People tell no. Anybody who ridicules any dimension of knowledge is ignorant. Alright, if somebody says science is more important, arts is not important, economics is not important, history is not important, what is more valuable is science, physics is more valuable than history. They are ignorant people. They don't know. I often hear people telling that you just have to memorize biological science. Biology is for memorization. You just have to memorize what it is. Dynamic logic is required. Dynamic logic is required for understanding life, because there are multiple parameters influencing the expression of certain things in life, and with the change of some parameters, a lot of things keep changing. So how can you come to a conclusion on what is happening and what needs to be done if you don't have dynamic logic? Okay. So don't being attached to the branch of study that you are doing is good, but ridiculing another branch of knowledge, another dimension of knowledge as being lesser is an indication of ignorance. That is the person who is not knowledgeable. Okay. If anybody says that, that is how I look at it. Everything is as important as anything else. Everything has its place, and out of its place nothing is important. Correct? No? If you are in a place where nobody understands English, you are as good as dumb. Correct? And you are as good as being deaf. Because what they speak you cannot understand, and what you speak they will not understand, then speaking and listening is of no use. So, you should figure out what is the place where your knowledge is valued, and you should try to be in that place. And you should get out of the expectation that your knowledge should be valued by everybody in every place. That will not happen. Whoever you are, in your place you are valuable. Outside of it, nobody cares. Don't expect people to care about who you are and what you know and what you do. Alright. So, am I useful or useless? Both. Both, no? I am useful in a place where my utility is there. I am useless in a place where I am not required. That's all. How you look at things will determine how well you grow. And you should be grounded. Yes, next. Oh, what is the answer to this question? We did not look at the answer only. The yellow color of urine is due to urochrome. Urochrome is a pigment. This one only. Urobilin only is urochrome. Sweating helps in removal of excess salt, regulation of body temperature, killing of skin bacteria, removal of excess water. Is there removal of salt during sweating? There is removal of salt. But is sweating a mechanism to remove salt? No. is there bacterial death that happens during sweating on the skin? Skin bacteria dies because of sweating. Symbiotic bacteria may not die, but external bacteria, pathogenic bacteria might die because of the high salinity that sweating causes. So, but sweating is not being, you're not sweating to kill bacteria. Okay? Water is removed, but the purpose of sweating is not to remove water. So, it is for cooling down the body temperature. Cooling down the body. How does it happen? It is because of surface evaporation by using the body heat. That is why exercise leads to sweating, and skin texture improves. Your skin improves because it is when you exercise, it is not only the sweat glands that are activated, even the sebaceous glands are activated. Sebaceous glands are also releasing their oil. Exercise is the best moisturizer. Cricket player is running fast to chase a ball. Which group of bones is directly involved? Okay. Pelvis, ulna, patella, tarsals. You should not consider the upper limb bones. We are assuming that the question is related to running. Running also, this is also happening, no? And is also moving. Actually, every bone is involved. But let us interpret it as leg bones. Femur, yes. Tarsals, metatarsals, tibia. Okay. What can you eliminate without any doubt? Sternum can be eliminated without any doubt, and malleus. Malleus is what? Ear ossicle. Ear ossicle, nothing is happening there. Okay. It might still be involved, cheering of the people, shouting of the bowler. Throw it fast. There was a cricket player. His son also played. His son was a batsman, maybe. Decent batsman. The father also was a good player. But in spite of his good batting skills and bowling skills, he was left out of the team because he was a very bad fielder. Any ball that goes to him, 1 run becomes 2 runs. Like that, 1 extra run if the ball goes to him while he was fielding. And more likely that he will drop a catch. So, and there is one player in the current Indian team, even if he doesn't perform with his bat, he is supposed to be a batsman, but he continued to be in the team because of his amazing fielding skills. Virat Kohli, not yet there. And he bowls also. This guy has become a mainstream bowler, main bowler also. Ravindra Jadeja. For a period of time, he was not performing only with bat, but somehow he can put some 4 or 5 overs. Within 2 minutes, he can finish 1 over. By the time batsman looks here and there, over is over. And he makes them waste the ball, builds pressure. Fielding, no, nobody dares to take a second run when the ball goes to Jadeja. Alright. Excessive, joints, 154. Which of the following pairs is correctly matched? Cartilaginous joint, skull bones, wrong. What is the joint? Fibrous joint. Hinge joint between vertebrae, wrong. Between vertebrae it is cartilaginous joint. Fibrous joint between phalanges. Between phalanges it is synovial joints, not fibrous joints. Gliding joint between zygapophysis of successive vertebrae. That is correct. Excessive stimulation of the vagus nerve in humans may lead to hoarse voice, peptic ulcer, efficient digestion of proteins, irregular contraction of the diaphragm. See, 2 of these functions are associated with vagus. 2 of the 4 options, vagus can do it. What are the 2? Vagus is a parasympathetic nerve, and vagus also has a motor component. Vagus supplies the diaphragm. So vagus, irregular firing of vagus can lead to irregular contractions of the diaphragm. But that is not the normal function of overstimulation. Vagus is a parasympathetic nerve, and parasympathetic stimulation on the gastric glands is that it increases gastric secretion. Digestive juices secretion will increase because of parasympathetic stimulation. Sympathetic stimulation makes digestive juices less. So if there is continuous overstimulation of vagus, hypersecretion of gastric juices, that means hypersecretion of gastric acid also, leading to gastric ulcers, peptic ulcers. Unresolving ulcers, one of the modes of treatment is vagotomy. Cut the vagus nerve branches supplying the stomach. So the vagus nerve branch supplying the stomach will be cut off to treat gastric ulcers. Ulcers are dangerous because once a gastric ulcer patient, you are permanently a gastric ulcer patient. They don't heal. Tremendously powerful acid. The acid will not allow the accumulation, build-up of mucus. Once the mucus is completely eroded, the acid will prevent the mucus from building up on the degraded part. So if not treated well and treated for long-term, gastric ulcer can lead to perforation of the stomach. Stomach perforation can happen. You take a drop of your gastric juice and put it on your skin, it will perforate. It is 1.5 pH. Very, very powerful acid. Homeostatic regulation of an animal requires three basic components: a detector, an evaluator, and an adjuster. Which of the following correctly matches these components? Detector is receptor. Evaluator is see, receptors, effectors. Receptor and effector. This is trying to conjugate as much as possible. So this is normal. There is no problem with this fellow. Whatever unconjugated bilirubin is coming, he is trying to conjugate it. But the problem is, there is too much of bilirubin being produced. Why? It can be because of too much of RBC destruction. You understand? This is the sequence. So, bilirubin is coming from him. Did you understand the story? That's all. So when you look at the blood report, bilirubin, total bilirubin, conjugated bilirubin, and unconjugated bilirubin, you can find out where the problem is. Total bilirubin is more, that means something is happening, there is more bilirubin. Of that, conjugated bilirubin level is normal or slightly raised, but unconjugated bilirubin level is very high. What kind of jaundice is this? Pre-hepatic, before the liver, hemolytic, breakdown of RBC. Conjugated bilirubin is low. Conjugated bilirubin is low, unconjugated bilirubin is high. Then, conjugated bilirubin is low, unconjugated bilirubin is high. Then what does it mean? Conjugation is not happening. Hepatic jaundice, hepatitis, inflammation. Unconjugated bilirubin is normal only or slightly raised. But conjugated bilirubin level is very high. Meaning what? Conjugated bilirubin is not getting excreted. Probably obstruction, gallstone, obstructive jaundice. Okay. People tell no. Anybody who ridicules any dimension of knowledge is ignorant. Alright, if somebody says science is more important, arts is not important, economics is not important, history is not important, what is more valuable is science, physics is more valuable than history. They are ignorant people. They don't know. I often hear people telling that you just have to memorize biological science. Biology is for memorization. You just have to memorize what is it. Dynamic logic is required. Dynamic logic is required for understanding life. Because there are multiple parameters influencing the expression of certain things in life, and with the change of some parameters, a lot of things keep changing. So how can you come to a conclusion on what is happening and what needs to be done if you don't have dynamic logic? Okay? So don't being attached to the branch of study that you are doing is good, but ridiculing another branch of knowledge, another dimension of knowledge as being lesser is an indication of ignorance. That is the person who is not knowledgeable. Okay? If anybody says that, that is how I look at it. Everything is as important as anything else. Everything has its place. And out of its place, nothing is important. Correct? No? If you are in a place where nobody understands English, you are as good as dumb. Correct? And you are as good as being deaf. Because what they speak you cannot understand, and what you speak they will not understand, then speaking and listening is of no use. So, you should figure out what is the place where your knowledge is valued, and you should try to be in that place. And you should get out of the expectation that your knowledge should be valued by everybody in every place. That will not happen. Whoever you are, in your place you are valuable. Outside of it, nobody cares. Don't expect people to care about who you are and what you know and what you do. So, androgens is given, that's still fine. What are the hormones associated with fetal ejection reflex? Or what is the primary hormone of fetal ejection reflex? Oxytocin. Oxytocin is required for 3 or 4 hormones. Endorphin, serotonin, oxytocin. These 3 hormones are very important hormones. See, endorphin is happiness hormone, makes you feel happy. When you start doing physical activity, how physical activity is related to state of mind. Doing physical activity every day, what it does? First, you start doing it for a few days, you start having endorphins being released, then serotonin, dopamine. Dopamine is the first one. Dopamine, endorphin, dopamine, serotonin, and oxytocin. Endorphin is happiness hormone. As you start doing exercise, dopamine is released, you start feeling good. You continue to do exercise for a few days, dopamine stabilizes. You will have constant levels of dopamine in your blood, and endorphins start getting produced. You will start feeling more happy. More happy means less stress, you will be more focused. Continue to do exercise every day for a few more days, few weeks, then serotonin starts getting released. Serotonin further makes you more relaxed. It relaxes and improves your state of mind. Reduces depression. Serotonin is given as a treatment for depression, for depression patients. It is produced naturally. Then continue further, oxytocin is released. Oxytocin is a hormone that will stimulate better self-image, better self-worth. Okay? Have you noticed mothers will be doing a lot of work, they will be going through a lot of physical stress, but they will be in a state of, they will be happy. Lactating mothers are happy generally. Oxytocin makes them feel good. So these 4 hormones will be stimulated when you exercise. Dopamine, endorphin, serotonin, oxytocin. Not on day one. You start exercising, dopamine release will start happening. Then gradually as you stabilize, endorphins will become more and more in bloodstream. Then serotonin starts picking up. Then oxytocin. So intellectual performance is obligatorily dependent on physical activity. So if somebody tells you you should completely sit and study only, you own nothing else, you should not do anything, you should not play, you should focus your every minute on studies, disaster that they are in. The job of guiding students for intellectual performance. And unfortunately, this disaster is over 90% of the education system. This is the disaster in the system. People who don't understand anything about how intellectual development happens are running the system and creating systems, creating big buildings, flashy things. People also don't know, they get attracted. Nonsense. But you know, you should stick to it. You can make note of those hormones. You can make note of this. This is life advice. You should study more of these 4 hormones. Do more research on these 4 hormones. Dopamine, endorphin. Endorphin is pain relief hormone also. It will reduce pain. Dopamine, endorphin, serotonin, oxytocin. Dopamine, endorphin, serotonin, oxytocin. These 4 hormones you should try to increase in your body. Figure out how these hormones increase and keep doing those activities. So playing every day, doing yoga every day is necessary. Necessary for a good life. Why I keep insisting is because we forget. Everything around us will tend to pull us back into the same pit that everybody are in. And every time you are in that pit, my voice should echo in your mind. This fellow has told that this is wrong, this is wrong, this is wrong, this is right, this is right, right and wrong, right and wrong. Should constantly get telling you. Whether you do the right things or not, you should know what the right thing is. Because at some point when it becomes necessary for you to do right things to get out of whatever problems you are in, you will know what is right. Okay. So I don't care about ridicule. You laugh at me, criticize me at the back of your mind. But what I make sure gets drilled into your head are the ones which will save you when saving is required. Next. Female hormone inhibin is secreted by. Inhibin is secreted by ovary. What of ovary? Zona pellucida of the ovary produces the mature follicle. The follicle is maturing. The mature follicle, zona pellucida will granulosa cells, not zona pellucida. Granulosa cells will produce inhibin. Inhibin will act on the anterior pituitary suppressing FSH. Inhibin has an anti-FSH action. So FSH stops, follicle does not grow further. When the follicle has grown sufficiently, FSH should stop. Inhibin is the one which gives that negative feedback. Okay. Granulosa cells of the ovary. And these granulosa cells are the ones which will produce estrogen also. Remember that. Head of the epididymis present at the head of the testis is called head. Caput is head, cauda is tail. So caput epididymis is the head of the epididymis. Cauda epididymis is the terminal part, tail of the epididymis. Gubernaculum, do you remember? Gubernaculum is the fibrous structure which will guide the descent of testis. Gubernaculum testis. In a woman after hysterectomy, removal of uterus, what happens to ovarian function? Stoppage of FSH secretion, no effect on the ovaries, complete hormonal imbalance, reduced estrogen levels over time. What will happen? Technically nothing should happen. Logically nothing should happen. Because ovarian part of the menstrual cycle and the uterine part of the menstrual cycle, uterine part is dependent on FSH, LH, estrogen, progesterone. You remove uterus, these 4 will not be acting on the uterus. But the ovarian part is dependent on FSH, LH. FSH, LH is still being produced. But in reality, estrogen levels will start going down gradually. Slowly estrogen levels will come down. If you remove ovary, if you do oophorectomy, ovary removal is oophorectomy. If you do oophorectomy, complete stoppage of estrogen right away. But if you do hysterectomy, gradual decrease in estrogen levels. Mammary glands are modified. Sweat glands. Mammary glands are modified sweat glands. Sweat glands are apocrine glands. What are apocrine glands? The gland remains and secretion is released. Holocrine glands are the cell of the gland is only removed along with the secretion. Sebaceous glands are holocrine glands. Holocrine glands, the entire sebaceous gland cell is removed along with the secretion. Weight loss, infections and cancers are common symptoms of AIDS, gonorrhea, vaginal candidiasis, genital warts. AIDS. See, AIDS progresses through stages. Acute infection, nothing much. There is a latent phase. During latent phase, the person might start developing on and off fever. More common infections are happening. And when it moves to AIDS-like symptoms or AIDS-related complex, then lymphadenopathy will start, weight loss will start showing, more diarrhea starts happening, gut infections, diarrhea starts happening. Full-blown AIDS will show opportunistic infections. One opportunistic infection you have to remember is one particular sarcoma of the tongue. What is that? Kaposi sarcoma. Kaposi sarcoma is characteristic of HIV. Alright? Pre-AIDS stage, the person will have chronic diarrhea, continuous fatigue, weight loss, all of this is pre-AIDS phase. Full-blown AIDS, opportunistic infections. Cancers, Kaposi's sarcoma. In IVF ET technology, the developing embryo is implanted at what stage? We have seen this. In vitro fertilization, embryo transfer. 8-celled embryo is transferred higher up into the uterus. In a cross between genotype AB and what is the other genotype? Genotype AB and what is given? What is given? 167. What?
In a cross between genotype AB
And plus plus, sir. Let us assume homozygous, recessive, or some other character. Small a, small b. Alright. 650 out of 1000 individuals were parental type. What is the distance between gene A and B? What is the unit for measuring the distance between genes? Centimorgan. Centimorgan. Centimorgan, 1% recombination is 1 centimorgan. 1% recombination is considered to be 1 centimorgan. Now, distance is recombination frequency is 650 out of 1000 individuals were parental type. So the recombination frequency is number of recombinants out of the total offspring as a percentage. 650 divided by 1000 into 100. 65% is the parental variety. 65% is the parental variety. 35% is the recombination frequency. 35% is the recombination frequency. So the distance is 35 centimorgans. Mutations which normally happen randomly are considered one of the raw materials for evolution because they contribute to new variation in organisms, cause the death of the organism, are stable, none of this. Mutations contribute to new variations and accumulation of these variations is evolution. Are mutations always beneficial? Mutations can be harmful also. Can mutations be fatal? Can mutations be fatal? Can they lead to death? And the other X chromosome becomes non-functional in terms of translating the genes present on the X chromosome. And that non-functional X chromosome is referred to as the bar body. So how many bar bodies will be found in males, human males? How many bar bodies can be found in human males? 0, because human males have only one X chromosome and that X chromosome has to be functional. How many bar bodies can be found in human females? 1. How many bar bodies can be found in Klinefelter syndrome? No. 1. Klinefelter syndrome is XXY. A male with 2 X chromosomes. One of the X chromosomes should be functional. So the number of bar bodies that you will find is the number of X chromosomes minus 1. Next. In the garden P, round seeds are dominant over wrinkled seeds. A plant with round seeds is crossed with another round seeded plant. 400 offsprings are produced. If one of the plants was heterozygous, how many offsprings will have wrinkled seeds? Round seeds are round. A plant with round seeds is crossed with another round seeded plant. 400 offsprings are produced. If one of the plant was heterozygous, that means the other one is homozygous. If one of the two round seeded plants are heterozygous, the other one is homozygous, no? How many offsprings will have wrinkled seed? Option is wrong. Options are wrong. If one of the plant is homozygous for round seed, that we can assume because they are mentioning that one of them is heterozygous. Both are producing round seeds. One of them is heterozygous. Then by consequence of that statement, the other one is homozygous. Then 50% of them will be homozygous for round and 50% of them will be heterozygous. All of them will be producing round seeds only. Option does not have it. Then what should we choose? What will you do if this is the question? The question is asking about wrinkled seed. One of them is homozygous, heterozygous is what is mentioned. You need to make an assumption for the other. And both are round seeded. So we have to make an assumption that the other one is also heterozygous. Then 50% of the plants will be with wrinkled seeds. Okay, this is a fit question for challenging. None of the options is actually right.